Now it's my pleasure to welcome Dr. Jan Lüddecke from Hahn-Schickard, Vision to Products, the slogan of Hahn-Schickard. And yes, we look forward to your presentation, Dr. Lüddecke.
My talk is focusing on liquid biopsy cancer diagnostics and using centrifugal microfluidics to unlock new cancer biomarkers. And especially I'm focusing on the use of extracellular vesicles in liquid biopsy. So I'm starting with a brief introduction into extracellular vesicles. So EVs, in short, are small particles that are secreted by all cells of the body. They have a double lipid layer and contain lots of different molecules like proteins and nucleic acids that can be used as biomarkers. The particles are quite small, so 30 to 200 nanometer in the range. Those are the ones that might be interesting for diagnostics. And yeah, as I said, they are released by all cells of the body and can be found in all body fluids. And they kind of reflect the state of their parent cells, and that could be cancer cells, but also immune cells. So the vision is that these EVs can be used as kind of a marker that reflects the state of the complete organism, basically. And because they are so abundant in body fluids, this gives them an edge over classical liquid biopsy markers like circulating tumor cells or cell-free DNA, which are usually low abundant in early stage cancer and MRD, and are also not shed by all tumor lesions. And there's also cancer-associated mutations in leukocytes, so that could lead to false positive results. Yeah, so liquid extracellular vesicles are really a promising alternative or maybe addition to these classical biomarkers. The field of EV research is quite young, as you can see here, the number of publications, but it has really seen a boom in the last years. And also the number of clinical studies has increased significantly during the last years. And most of these studies actually focus on using EVs as diagnostic tools in cancer. And lots of, or the majority of these studies focus on EVs from blood, plasma, or serum. But despite yeah, these big expectations and this promising basics here that I showed, EVs haven't really reached the clinical application yet, at least not in a big scale. And the question is, why is that the case? And if you look at the publications, you will see in a lot of reviews that one of the big issues in EV research is actually the EV purification. So there's a lack of standardization. There's hundreds of different methods that are used, and many of them are complex manual processes and not really easily transferable into clinics. And this also leads to results being not really reproducible. And that's, of course, a big problem. So from our point of view, we see a big need in the field of EV research in a standardized purification method that automates the purification, ideally starting with a blood sample, that minimizes user influence and that combines different purification modalities, because as I will show in the next slides, to get rid of all the contaminants or background, you need to combine different methods. And of course, the method needs to provide sufficient amount of EVs for downstream analysis. And such a method then would enable reproducible clinical studies and is, in our point of view, mandatory for clinical applications. And of course, this is what we are working on. But first, let me show what's actually making it so challenging to purify EVs from blood. So of course, there's lots of particles in blood, and especially the lipoproteins, the high density lipoproteins and low and very low density lipoproteins are critical because they are in a similar size compared to the EVs. And as you can see in this picture, this shows the concentrations. EVs are concentrated in a, yeah, in the range of 10 to the power of 9 particles per milliliter, and these lipoproteins are concentrated a lot higher. So that's one of the big challenges if you want to use EVs for diagnostics and use blood as a source, then you need to get rid of all this background to be able to analyze EVs. And of course, you can purify EVs by using different properties like the density, the molecular composition, size, and charge. But many of these methods are not really suitable for clinical application. For example, yeah, ultracentrifugation, big bulky instruments takes a lot of time, not really compatible with routine diagnostics. And also molecular composition, there's no universal EV marker that you could use to capture EVs. So unless you know exactly what you're looking for, immunocapture is also not really the way to go. So in our approach, we're focusing on size and charge to purify EVs. And yeah, just as an example, with these two methods, you can also get rid of most of the lipoproteins. So you can see the charge of the particles, the low density lipoproteins up here are positively charged, EVs and high density lipoproteins are negatively charged. So with a charge-based purification, you can deplete most of the LDLs, and then if you apply a size-based separation, you can also remove the smaller HDLs from the EVs. So the workflow that we try to automate and use for EV purification has three main steps. In the first step, we use a cation exchange resin, so negatively charged particles that bind the positively charged LDLs. The EVs are not held back here. And then in the second step, we have actually two filters that we integrated. One with a 1 micrometer pore size and 20 nanometer pore size, so we can get rid of bigger particles like platelets and cell debris, and also smaller particles like proteins and HDLs, and concentrate our EVs. And then we have kind of a final polishing step where we again use a multimodal chromatography resin to remove remaining small proteins. So this process looks pretty complex and it's lots of different steps. And as I mentioned before, we need to standardize and automate. So what we are doing here at Hahn-Schickard, one of our core technology is actually centrifugal microfluidics. And that's what we are using to automate this purification process. And we call our cartridges lab discs because they look like little CDs or DVDs. And we use centrifugal forces to push the liquids through our channels here and automate all kinds of biochemical assays on these cartridges. And here's an example application from our spin-off company, diagnostics. They have a cartridge that automates a skin disease diagnostic assay. Yeah, and these cartridges are ideally suited for point-of-care applications. And this is our prototype for vesicle purification. We call it the V-disc. It's an injection molded cartridge, and you can see we have integrated here the two filter membranes that I mentioned before. And we also can pre-store the chromatography resins in the cartridge, and then upon addition of a resuspension buffer under the centrifugal forces, we can create these little chromatography columns on our cartridge that we use then for purifying the EVs. And here's how the cartridge looks after the process. So you see the resin column here. You see there's the plasma and the waste liquid, and then you can pipe it out the EVs from this chamber down here. And coming to some results, so we compared our disc to a conventional EV purification method, which is size exclusion chromatography. Lots of labs use these ISON columns to purify EVs. And regarding EV recovery, we used fluorescently labeled EVs, and we get a recovery of about 84% with our disc compared to 60% with a size exclusion chromatography. The second graph shows basically the ratio of signal to noise, so EV signal divided by protein background, and we tested different plasma volumes on our cartridge from 1 ml to 100 microliter, and you see the best ratio of EV yield and low protein contamination is achieved with 500 microliter of sample. And we also tested removal of the lipoproteins. So with our cartridge, similar to the SEC, we can remove up to 99% of the high-density lipoproteins and more than 93% of the low-density lipoproteins. But here the SEC is still a little bit ahead. Yeah, and some particle analysis results here. So as I said, EVs that are interesting for diagnostics are in the size range of 30 to 200 nanometer, and this is also the size range of particles we purify with our cartridge. And 10 pictures also show that the EVs that we purify on our cartridge are intact. To automate our centrifugal microfluidic cartridge, as I mentioned, we use these instruments like the lab displayer, which we have developed. It's a small benchtop device that can handle one disk at a time, but we are also working on improving the throughput by integrating these lab displayers into a pipetting robot. So this robot can automatically load the disk and inject the sample and so on. So to summarize, with the current version of our vesicle disk, we can automate the purification of EVs by charge and size from 1 ml of plasma, and we combine different purification modalities, and the disk outperforms the standard method of size exclusion chromatography. The next steps will be to fully integrate the cartridge with all reagent pre-stored and starting also with plasma separation. So our aim is that we can load directly blood onto the cartridge, so we don't have to transport any plasma. And yeah, exactly. And application scenarios would be clinical studies, of course, where the need for standardization is very high, and this cartridge would enable comparability between different clinical study sites and trials. But of course, in the long run, the disk could also be used for diagnostic tests. If we integrate a EV analysis onto the cartridge, we could have a sample to answer system basically. And we are actually currently working on such a system with our clinical partners in Erlangen in the BMBF project Key West, where we aim for prostate cancer monitoring, and we use EVs purified by our cartridge combined with an AI pattern recognition model to monitor prostate cancer. Yeah, we're open for cooperation to bring our technology into the market. And with that, I would like to thank my team, mostly Esan and Gustav, our fluidic engineers who developed the cartridge, and of course our cooperation partners in Erlangen and also here in Freiburg for their support. Thank you very much.
Thank you so much, Dr. Lüddecke, for this inspiring talk. And thank you so much, Sarai, from the audience for an interesting question. So she's saying, very interesting application of microfluidics here. If I am interested in proteomics, I would also like to classify the EV contents, e.g. into different types of RNA. Is this possible?
Yeah, we haven't tested it yet. We haven't worked with RNA markers yet. But of course, it's a very interesting field of application in the EV field. MicroRNAs are very important markers. And it all depends on how pure your EVs need to be. But I think with our method, it's probably pure enough to study microRNAs.
Very interesting. Thank you so much. No other questions from the audience. We have some minutes. I have one question, very simple question. So, because I'm not an oncology doctor, but if I got you right, the extracellular vesicles are released by all cells.
Yeah, that's
so
true.
How can we deal with the background noise in an effective manner? Do you think your methods will be suitable, or when will they be suitable for routine and daily cancer screening?
Yeah, that's a very good question, of course, especially in early cancer when you just have a couple of cancer cells, you need to find the needle in the haystack, basically, and that also doesn't change with EVs where you have all the background. So in the project with our partners in Erlangen, we're focusing on enzymatic activity of EVs, but we also take into account the immune response. So we don't only look for EVs that are released by cancer cells themselves, but also by the immune system, and trying to combine this method with the help of AI and pattern recognition to get a clear picture on what is really happening. But that's true, that's probably one of the biggest challenge in early cancer detection to distinguish between background.
Thank you so much, Dr. Lüddecke. Truly fascinating topic.
Thank you.
Thank you once again. Thank you to the audience. No further questions. Thank you so much.